Thank you so much, and thank you as well for the invitation to speak today and present some of the work that our group and collaborators have been doing for the last few years, trying to bring together data sets from human islet preparations that we do here in Edmonton, and essentially trying to produce a tool that allows this data to be used and explored. I'm just looking at the attendees, and I want to give a shout out to Jess Ewald, who is here, and Kara Ellis, who is here from our team and from our collaborators. Jess gave a version of this webinar previously, so I've stolen a bit of it from her. So I hope she'll speak up if I mess up any of her slides. Jess, I will say, has been instrumental in building the web tool that I'm going to show you today. So I'm going to share my screen here, and we're going to start with some slides. I want to encourage people to speak up if you have any questions. I may not be able to hear you. Well, I should be able to hear you. I may not be able to see your hands raised, but I'm happy to take questions as we go through, or at the end as well. So I'm going to be talking about a tiny bit about our human pancreas program called the ADI, the Alberta Diabetes Institute IsletCore. Mostly I'll talk about the web tool we've put together at humanislets.com, and I'll take you through a few bits and pieces in a live demo of what this tool can do. Excuse me. So when we started our program for human islet isolation back in sort of over time, from about 2012 through 2015 or so, we were building this human research program. There was some question in the field about accessibility to human tissue, particularly human islets, for diabetes research. And there was a keystone meeting at the time that raised some concerns about access to islets, and that really is what spurred us to try and expand our efforts in human islet biobanking and research. There are still issues that are brought up from time to time about the quality of research, quality of data, quality of tissue, and so we have more to do in the human research islet space, and that's one of our main interests for our group. Well, this sort of messed up here a bit, but what we started back in 2011 was called the Alberta Diabetes Institute IsletCore. This is a research-only program that essentially takes pancreas from organ donors when it is declined for clinical use. So if an organ is not going for clinical transplantation or for islet isolation and islet transplantation, then it can come to us with research consent, of course, for our research program. We isolate islets from a broad range of donors, young, old, with and without type 1 diabetes, type 2 diabetes. We do about 50 to 60 organs per year, and we work now with islets, but also other associated tissues like whole pancreas, of course, but also spleen, pancreatic lymph nodes, adipose, gut, blood, and plasma. I'm going to be talking about islets only in this talk. So we have quite a large network of users of this biobank across the world, about 160 or so groups and institutions that access either fresh or banked tissue from us. In this time, we've processed 557 donors. It's actually, I think it's up to 559 now as of this past weekend. We have a large number of islets, or islet equivalents, we call it, banked. We are proud of the fact that we are sustainable and we do not work on any subsidy. We rely entirely on cost recovery charges. We have open protocols at protocols.io. We publish on our website things like this user guide to human islets, which is shown at the right. And we can do collaborative work like custom sample prep or live pancreas slices. We often have visiting scientists in the lab. We have a couple here from Germany at the moment. So, to get to talking about our web tool and how we brought data sets together, it's important to talk about collaborations. So one of the most exciting things about running this program has been the ability to become involved in all kinds of different research projects. And I'm just showing one here because it's relevant to the web tool. This is a paper published earlier this year by Yelena Kolick up at the top here in Jim Johnson's lab, where they are phenotyping islets from our islet isolation program and looking at whole islet proteomics together with dynamic insulin secretory responses to different macronutrients like glucose, amino acids, and fats. And then they are correlating, proteomic protein expression with secretory responses. Here I'm just showing up in non-diabetes and type 2, but if you look at the paper, there's all kinds of data linking, for example, insulin secretion response to fatty acids with the protein expression profiles of those donor islets. And we have a lot of these kinds of collaborations across Canada and North America, bringing together, looking at different types of data. With Anna Gloin, for example, we have ongoing projects in genetics and genomics. So all of our donors are genotyped, for example. We have RNA-seq and ATAC-seq on all of the donors and so on. So out of all these collaborations came this idea to try and bring together these disparate data sets from the same donors through what you might call a distributed phenotyping network. And here are the individuals at the top who are involved. And our protocol or algorithm looks like this at present. So we receive a whole organ here in Edmonton. Of course, we collect the metadata from the donor, but also samples from the donor, like plasma samples, for example. We process that organ to isolate islets. This is a nice islet prep from a human donor. We, of course, collect data on the processing and all of the different parameters for the isolation itself and isolation outcomes. We collect biopsies and other tissue samples and other samples, for example, environmental toxicology. We culture these. We have data from cell culture and QC, and then we phenotype. And these are the phenotyping assays that are commonly done now on all of our islet isolation preps: insulin secretion, gene expression, ATAC-seq, RNA-seq, electrophysiology, which is my background. Our group does electrophysiology on everything. Patch-seq was mentioned earlier. Oxygen consumption measurements, dynamic insulin secretion. So we're collecting all of this data, and we've been working for the past couple of years to put this together in a web tool. Again, a big shout out to Jess, who's shown at the top right here, who effectively developed the entire web resource that I'm going to show you today. She was working with Jeff Shaw at McGill and is now at the Broad. So what is in this tool at present? Right now, there is data on 547 donors with 74 metadata variables and 5 different types of omics data. We really want this to be as broadly usable as possible. I am not a computational biologist. I'm not a geneticist, or I don't work very much in genomics. I find a lot of tools that are out there with human islet data are challenging to use for someone that doesn't have that kind of background. And so I really wanted us to try and build something that could be as broadly usable as possible. Also wanted it to be open and transparent. So everything that I'm going to show you shortly is downloadable, including all of the analysis pipelines, so that things can be replicated. This was funded through a team grant. So that phenotyping network was brought together by a team grant supported by the Canadian Institutes of Health Research, Diabetes Canada, and JDRF Canada. And if you look at the tool, there are several ways to go through it. I'm going to show you this live shortly, but there are essentially three different sections to the tool: an omics view that lets us look at pathways, a feature view that lets you search for specific features like a specific gene and see what it's correlated with in our database, and a donor view, which lets you look at data for each individual organ donor that we've processed. One of the really cool things that has come out of this this exercise and bringing all this data together is the ability to look at how different metadata or different properties of an islet isolation may impact the omics data that you're looking at. So a clear example is the effect of culture time. And it just did quite a lot of work looking at this. Kara also did some work looking at culture time. But really what it boils down to, from the way I see it, is that culture time has a strong impact on lots of transcripts in bulk transcriptomics of islets. And you see that on the left here. We're looking at FGF2 transcript in this case, going up with culture time. We can confirm that in a separate assay, a nanostring gene panel, where we happen to have FGF2 in there, going up with culture time. But protein levels seem to be much less affected. And possibly that's just because of the timing involved. If we went longer, maybe we'd see changes in the protein. But I'll show you how we can correct for some of these things in the analysis on the web tool. Well, actually, sorry, I'll show you it in practice, but here's a result where Jess has corrected for culture time with RNA-seq data, essentially showing that adjusting for culture time decreases, I believe, the p-values for a number of the hits that you might see. The other thing that was really interesting coming out of this is cell type analysis. And we look at this in a couple of different ways. This is a single cell RNA-seq data set put together by Kara, shown at the right, where she has identified a number of islet and pancreas associated cell types. And so I'll show you again on the tool where we can explore this. But what became apparent looking at a large number of islet preparations is that percent purity of the prep is actually quite important in the analysis. And I'll show you what I mean. Well, I'll see if I can show you what I mean. When we do most of our assays, we have an islet prep that is, let's say, 80% pure, meaning there's 80% islet tissue, 20% acinar tissue. When we do proteomics, for example, we handpick the islets to 100% purity. Well, if you look at the data, it turns out that you really can't pick islets to 100% purity. There's probably acinar and other tissue hanging on to those islets even after you pick them. So when you are picking islets to 100% purity, if they come from a prep that is of lower purity, you're still going to have some acinar tissue in there, and that tends to impact some of the results. This becomes important comparing, for example, non-diabetes donors with type 2 diabetes donors, because islet preparations from donors with type 2 diabetes tend to be much less pure. So Jess, in thinking about how to deal with this issue, and this will connect back to the cell type analysis that I mentioned. But what Jess did was implement a deconvolution approach to the proteomic data in order to estimate cell type proportions. And so you can take the proteomics, and using a set of known marker proteins, you can estimate cell type proportions in the islet preps that have proteomics data. Cell types look a little something like this. And across many donors, I think there's about 120 here, you can see that there's quite a bit of variation in the amount of acinar tissue, or I guess non-endocrine we'll call it here, and beta and alpha cells, from the low end to the high end. So the proportion of non-endocrine, for example, can correlate quite well with known markers of non-endocrine tissue in pancreas. And this can be corrected for now if you use that deconvolution data, adjusting for the proportion of non-endocrine cells. So you can see, looking in this case, a number of hits are reduced increased in p-value or decreased in significance when you correct for that, probably because some of those hits were coming from acinar tissue that contaminated the sample. All right, so I'm going to go to the website now and do a bit of a demo. While I'm switching over, are there any questions? All right, I hope I got all that right, Jess. I'm just going to switch my screen sharing here. Oh, that's the dkNET site. But there's our website. Can everyone see that? I hope that's coming across okay.
Yes.
Speak up if it's not. So this is the web tool. If you go to humanislets.com, you can find all kinds of information on the tool itself and how we put it together through the documentation. I'll just show you that really quickly. It's a pretty extensive document talking about how we, for example, did proteomics and so on, with links to the relevant papers for that. There is information about our team up here, and again, the publications that underlie this work. So I mentioned that there are three different main sections for the website. Actually, we should probably say there are four. So there's the omics view, donor view, feature view, but there's also a page for downloading data, which you can access here or here. I'll show you that in a moment. But we'll start in the donor view. So if you click on the donor view, it brings you to a page like this. This is donor R126. Each of our donors has an RRID, I'll point out, so you can link them to publications and so on. This donor in particular, which just sort of came up at random, has no diagnosis of diabetes, and it's a male, HbA1c of 5.2. You can see in relation to all of our donors where that stands. 38 years old, a bit on the youngish side, with a BMI of 24.1. On the left-hand side, you can click through pages with all kinds of different information. So this is info on the donor. This is the result of the islet isolation. You can see on the right, it's not the most pure isolation in the world. There's pictures from after the isolation, and well, if we sent them out, there will be islets for pre-distribution. I'm going to try to find another donor here. There was one I was looking at the other day. Let's try donor 246. Search that donor. There we go. So now this donor came to us with no diagnosis of diabetes. You'll see here it's adjusted to pre-type 2. So we actually have two different indicators of diabetes status in the web tool. The first is what we are told when the donor is sent to us. So this donor, say no diabetes, but we classify donors afterwards as well based on HbA1c. So anything greater than 5.7 would be considered pre-diabetes. Occasionally, for example, we'll get a donor with no diagnosis of diabetes, but they have an HbA1c of 7, let's say, then this adjusted status would be type 2 diabetes most likely. On the right here, you can see the inventory we have. We have 4 cryopreserved tubes of this donor in case anyone wanted to follow up and use tissue from this particular donor. So the isolation information is shown here. You can click through all the different properties of the isolation outcome. Pictures I showed on the right here. In the cell type composition, because this donor had proteomics done, we have the cell type composition at the bottom. We have phenotyping data here as well. So we have static glucose-stimulated insulin secretion. Again, you can see where the donor lies in relation to all the other donors with insulin secretion, stimulation index, culture time, insulin content, and so on. With Jim Johnson's group and Yelena Kolick at University of British Columbia, we do dynamic insulin secretory responses. So here's in the black is this particular donor, R246, and where it stands in relation to all the others. This is in response to glucose. You can normalize that data to the baseline. You can look at the area under the curve for all these different sections shown at the bottom here. And you can look at responses to glucose, to leucine, or to palmitate. More recently, we've started to do mitochondrial oxygen consumption measurements with seahorse assay. This particular donor does not have it, but here are all the donors that are in the tool so far. You can again normalize that data, and you can look at here again different properties of the respiratory activity of these islets. So if this donor was done, you would see a line here that shows where it stands for, in this case, maximum glucose-stimulated respiration. Finally, we do electrophysiology on everything, so you can look at total exocytosis of beta cells from each donor, or you can flip over to alpha cells, for example. And there are different electrophysiologic measurements done at different glucose concentrations. So that's it for donor view. And really, the purpose of the donor view is to get, if you want to look at a particular donor and see, how that donor's islets behave and what the phenotyping looks like and what exactly was done with that donor, you can use this tool. You can search by RRID up here as well, by the way. But other than viewing this, there's not a whole ton that can be done analysis-wise on this page. And you see, I'm looking at the chat here. Ashley asks, the omics data of a single donor. Good question. You can, if I click on this omics overview, it will tell you what's available for that donor, but you cannot view, for example, the proteomics for that specific donor. I'll show you how we look at proteomics in 1 second. You can download, by the way, the omics data, the processed omics data for individual donors, and I'll show you that in a moment as well. I'm going to go to the omics view. So to do some analysis on the data that we've just looked at, you can use this page here. And what it allows you to do is analyze our omics data. And you can see we have bulk RNA-seq, bulk Islet nanostring gene panel, bulk proteomics. We have some pseudobulk gene expression for alpha cells and beta cells. And we have some single cell data we can look at with the patch-seq, and maybe I'll come back to that a little bit later. But what I'll show you is we'll look at gene expression, and we will look at how islet gene expression changes with diabetes. Okay? There are all kinds of different metadata that you can analyze here, but for the moment, we will just look at diabetes. And we'll compare type 2 diabetes versus no diabetes. We talked a little bit about culture time, so we'll add culture time as a covariate, and maybe we'll add age and sex and BMI as well. Okay. You can select which donors you want to include in this analysis, right? So we're going to analyze type 2 versus no diabetes in this case. I'm not going to filter any donors here, but that's how you could do it if you wanted to. Just look at non-diabetes donors, for example, and you can adjust the p-value cutoff. Then you click submit. You should get it's coming. Okay, we'll start that again. RNA-seq diabetes. And I see there's a question from Kristen in the chat. Oh, there. So the analysis went through. But before I scroll down, I think this is a good question for Jess, maybe. I do believe that correction for multiple testing was done in this analysis. Jess, is that correct?
Yeah. Well, first of all, you see there's that little checkbox right there that says use adjusted p-values. So that's for, the overall summary that you get, whether you want to use the threshold on the raw p-value or the adjusted. And then in the results table, we always return the adjusted p-value, and it should be right there. And then in our kind of pre-computed analysis, that's all using the adjusted p-value.
Okay, so I guess my question is that adjusted p-value just based on this specific test or all possible tests that you can do on this page?
Well, in that analysis that Pat just submitted, it would have, so it's done at an omics layer at a time. So in this case, he analyzed RNA-seq data. So it would have been and then he did that specific comparison. So it was adjusted for anything that was analyzed here, but it's not like considering every single possible test you could have done across like the proteomics and the other transcriptomics types.
It's not global.
Yeah.
Cool. Thank you. I appreciate it.
Great. Thanks. Happy for suggestions too. We're continually developing this. I think Yao Lu, I don't think she's here, but is working on this now. So happy to hear your thoughts. So doing this analysis, we come back with results that look like this. We can click on a hit and look at the normalized protein abundance in this case, comparing type 2 diabetes with no diabetes. This can be exported as graphics, as graphic or as CSV file. If we look at the results table, there are a number of things that can be done here. So here are the results sorted. I believe they're sorted by adjusted p-value to start. You can change that and adjust by log, twofold change. There are a number of interesting things you can do on this results page. You click here, it'll bring up that same graphic showing you, in this case, islet amyloid polypeptide IAPP is down in type 2 diabetes, which is a good check. IAPP known to be important in diabetes. We can link out to the NCBI page, the description of that there. We can link out to the type 2 diabetes knowledge portal, which lets you explore whether there is genetic evidence, for example, for a particular hit. I think my internet's a bit slow here today. Here we go. Takes a second to populate. Right, so you can look at huge scores for diabetes, associations, and so on. But that's really just a link out to another resource that lets you learn a bit more about what you're looking at. Kara built this meta-analysis of single-cell RNA-seq data, so you can look at your target of interest from these results and see at the transcriptomic level which cell type is expressed in. IPP, well known to be enriched in pancreatic beta cells, as shown here. And you can go from here to pathway analysis. Oh, I should mention, all of these results are downloadable. The analysis also downloadable. So if I click this analysis, it comes. I'm not sure you're going to see this, but I'll share that screen. So I want you guys to see what it looks like. 1 second. There it is. So here's the analysis, and you can get the whole pipeline for that analysis here with the download. All right. You can go then to pathway analysis, and you can take those results and put them right into an overrepresentation analysis, adjusting FDR cutoff, deciding which gene set library you want to look at. You can do ORA or gene set enrichment by log 2 fold change or T statistic. We'll see what happens here. And you get results that look like this in a nice ridge plot that just put together. So some things that make sense in type 2 diabetes, lots of inflammatory and immune things, some metabolic pathways, and so on. Often you'll get a lot of pathways. You can rerun this with this collapse redundant sets box checked. Of course, it goes slowly when we're doing a demo, but there we go. So you have all of these pathways that come up. You can look at the results, download and save the analysis. You can also click on these guys to get a heat map and see which features are popping up in that pathway. The stars here denote the ones that came up as significant in that initial volcano plot. You can download this again as well. All right. Now, if we go back to the results table here, so that's the omics analysis. And I'm not going to go through it. Maybe come back to it if we have time to look at some other things. But I would suggest if you're interested, have a look through all the different things you can do. You'll find that in some cases, some things may be a bit underpowered. So we have in this RNA-seq data set, we have 120 donors. In the next version that's coming, we're going to be adding maybe 250 or 300 more donors to that, and hundreds of more donors to the proteomics. So this is really going to grow in terms of how much data is available for analysis. So I showed you the donor view. You can access them up here on the left, and the omics view. There's another one called feature view, which is interesting if you want to look at a particular feature of interest or a particular gene across data sets. You can get there by clicking on feature view, but if you have a list of hits here, you can also get there by clicking on the protein name in this case. And so that will take you to the feature view and give you the results for the feature view search for that gene or protein of interest, or transcript or protein of interest. And so what you're looking at here is a list of pre-computed associations for IIAP across all of our data sets. So it seems to correlate with the size of beta cells, which is something we measure in our electrophysiology. It correlates, again, with type 2 diabetes status, either corrected or uncorrected. This is in the nanostring data, so this is not the RNA-seq we were looking at before, so that's nice. Here's that association we were looking at earlier, this IPP being downregulated in type 2 diabetes, associated with beta cell proportion and non-endocrine cell proportion, associated with measures of insulin secretion like stimulation index. So we can click on this to get a scatter plot, see what that looks like. If we can sort these or search, so we know that IPP is down at the transcript level. Maybe we want to look at the protein level. We can protein. So bulk protein expression, it seems to be, where'd that go? At least nominally down as well in type 2 diabetes. So here's the protein from the proteomics, IPP, maybe down. We suffer a bit from having not quite enough type 2 donors as well, but that will change over time. You can search for a gene of interest as well. So I'm an electrophysiologist. I might be interested in ABCC8, which is the sulfonylurea receptor. So we can search for that. And we see all kinds of things pop up here as well, like type 2 diabetes, beta cell proportion, measures of insulin secretion, for example, so on. And that can all be downloaded. Right. So the last thing that I'll take you through is the data download. So one of the people, I forget your name, sorry, who asked the question just a second ago, asked about, how the analysis was done and how things are set up on this page and these pages and so on. And, this is really set up for fairly straightforward exploration of what we have in our database. There are, of course, people who are more computationally and statistically savvy than me, who may want to take the data and look at this themselves and do their own analyses. So the data download is really set up to do that. So if you click on data download, you come to this filter, which is very similar, if not the same, as the donor selection for the analysis on the last page. But you can decide here which donors you want to get data from. So we can filter, for example, only female donors. only female donors without diabetes by unclicking those. We can look at the distribution of these donor types here. So we have, not quite 500 donors without diabetes and less with type 1 and type 2. And we can filter by age, BMI, HbA1c, and lots of other things, like whether we have any inventory. You might want data only from samples that we have here in our biobank available for further experiments. Can also filter by data availability. So maybe you only want data from donors that have electrophysiology or something like that. But what we'll do is we'll apply this filter. We'll see there are 194 donors that meet that criteria. You can download that list of donors. And the person who asked about omics data from one particular donor, I guess what you could do actually is go to this donor list. And if there is a donor you want, you would just put in, right, R246 and submit that. And that would give you a single donor, which you could download data from. But I'm not going to do that right now. So we've got our 194 donors. We go to the right. We can look at the different types of data that we want to access and download. So all of the donor and isolation metadata, all of the insulin secretion data, electrophysiology, oxygen consumption measurements, nanostring, gene expression, proteomics, RNA-seq, so on. I'm going to select, see the donor info, and I guess nanostring gene panels. I guess just a quick note about the omics data. And Jess can comment a bit more on this. She's the one who put much of this together. So the raw omics data is not housed here. For the most part, that is housed in standard repositories, such as Geo, for example. So what you'll get here are count tables that come in two different flavors. One which we call, what do we call it, Jess? We call them, might be called corrected and uncorrected. So in some cases, like the bulk RNA-seq, we'll provide it with batch correction and without batch correction and normalization. Do I have that right, Jess?
Yeah, I guess we were like, it wasn't feasible to host the raw FASTQ files, for example, on this tool because that's just really hard in a web-based tool. And we wanted to make the data sort of as useful as possible to people who are maybe computational researchers and wanted to implement the whole pipeline themselves. So we call this like minimally processed or unprocessed because it would be just the very first matrix that you get after processing the data.
Right. Great, thank you. So you can select these things. You can choose what format you'd like them in, CSV, text files, R, or Excel. Because, let's see, I'll just choose text. You can look at a summary of the data you've chosen. So we've chosen all female, so on the right here, all non-diabetes on the right, and then this is just the distribution of donors in what we've chosen in the orange compared to the whole data set. And then you can download. And then I'll show you what that looks like. Stop sharing. So that was that analysis earlier. This is what the data looks like. So nanostring, unprocessed, and processed, and the donor info that I requested. And stop sharing that. And And that's basically it. I can show you more about the web tool if you like. Please feel free to play with it and ask questions. I'll go back to my slides very briefly. I don't think there's anything further really. The site I mentioned is continuing to grow. We have other data. We have many more donors to add in the not too distant future. We have other data types that we are collecting and will go into the tool eventually. We have lots of imaging data that we're trying to figure out how we can incorporate, and we're happy for feedback. And that's that. There's the team again. So I'm happy to answer any questions. I hope that was informative. I'm glad that Jess was here. But yeah, any questions?
Sure. So now is our Q&A session. You're welcome to unmute yourself to ask question, or you can also type in the chat. I think, Patrick, you already answered Ashley and Kristen's question during your presentation, right?
Yes. I guess what I'll add to that is that we are, and Yao, who's doing this, is not here now. So we are working on multi-omics analysis for this, which may also address some of the, some of your thoughts in that question, Kirsten. I see Ashley has a hand up as well.
Yeah, Ashley has a hand raised. Yeah, Ashley, you could ask question now.
Yeah, thank you so much for your presentation. This is a very useful resources for the community. I just wondering, sorry, I didn't see clearly when you download the omic data for particular, when you subset donor, there are different RNA-seq data. Some are from pancreas islet, some are from blood. Do you have those also? Can you subset?
Sorry, so omics from first of all, we don't have any omics from blood.
Okay. It's a proteomics from plasma.
No, we have nothing at the moment from plasma. What we do have are banked plasma samples from most of the donors. And the original idea when we put in this grant was to measure hormones, like particularly sex hormones, because that may be important as well to some aspects of islet function. But we could measure other things as well from those samples. So, we still are figuring out exactly how to use those in this particular tool.
Yeah, because it's going to be very difficult in a clinic, right, to do some of those islet kind of
of course, yeah,
phenotype. But it is easier to to assay the blood. I'm just wondering if there's some data from the blood, then find some biomarkers that correlate with the islet phenotype.
Yeah, that's a great question. I think one of the things that could be done, I guess we could do proteomics or metabolomics perhaps of the blood plasma, and then look at the correlation of that with islet function outcomes in the tool. I think that would be actually quite interesting. The other possible approach would be to, really the tool itself is, I think, quite useful for hypothesis generation. And so perhaps we'll come up with or someone can come up with some ideas of things that we want to go back and measure in our blood samples or other bank samples that we have as well. I think that can be done.
Yeah, if I may, have you think about, how to structure the download easier for pipe into AI algorithm?
A great question. I'm not sure that I'm equipped to answer it, but I will say that Jeff Shaw, who runs the group at McGill, where Jess was working, who built the tool, are thinking about that right now. So whether there are some changes needed in the download itself, I think really he's keen to incorporate some AI tools into this web tool itself as well. So yeah, we have some thinking to do on that. I think it would be interesting.
Thank you.
Yeah,
Really
thank you.
presentation.
Thank you, Ashley. So there is a comment in the chat from Katie, outstanding tour for our community. And thank you and congratulations. Yeah. And
Thanks. Oh yeah, Yassi has a hand up.
yeah, and there is another question from AB. So where is and how to cite Islet?
There's a link that Jess has put in the chat, which takes you to the what's called the terms of service. But there's citation and acknowledgement information on that link.
Is that answer your question, or do you mean the islet or the donor?
Yeah, if there's more to that question, feel free to ask. I will say too, although I think the link is now on the site, but you know, this was published just a few weeks ago in Cell Metabolism, Jess's paper. So you can cite that too.
Okay, great. And do you have any of the islets being cited by RID yet? Do you push for that?
I'm not sure what you mean. Islets cited by RRID?
Do you have RIDs for these?
Yes, we do. We do. They can be searched on the site by RID, and when we send so we do a lot of shipping of islet tissue to our researchers. We always provide the RID. As seems like you may be aware, but it sometimes is hard to get them to use it.
Right. So sometimes it's nice to have that listing also on the main page where you're asking people to cite stuff.
Okay, yeah. Yeah, I think that's a good point. And actually what we could do, here I can share the screen again. So this is the citation page, right?
Mm-hmm.
We could ask them to cite the RRID, but another thing that comes to my mind is on the data download. When they download a list of donors, this list is actually our internal donor number, but we could include our RRID there as well, so that they have access to it there.
Yeah, I think that might be better because, you know, one of the issues we've always struggled with, or we've struggled with for the last couple of years anyway, is you know, people know to cite their RIDs for their antibodies. They're pretty good at that. They're actually pretty good. Actually, they're getting much better at citing core RIDs. But and cell lines are pretty good, but the biosamples RIDs are spotty and sporadic. And so I think there needs to be a little bit of pressure in sense of you know, just at least some clarity on where to put the citations and how to use them. Because it you know, I know that people are trying to do the right thing a lot of times. And so if there's just a lot of clarity around it, it could help.
Yeah, that's helpful. I'll pass that along when we are, you know, as we are developing the next version of the site, which is ongoing now.
All right, great, thank you.
Thank you.
We have one question from Yassi. Yeah, Yassi, you can unmute.
Hi, Yassi, I can't hear you.
Yeah, I can't hear you.
Can you hear me now?
Yes.
Okay. Hi. Yeah. Yasi couldn't be here, so it's just me using her link to
okay. Yeah.
attend the session. But thank you very much, Patrick. This is an amazing resource. There's a lot of information in there. Congratulations to you and your team. I was just curious. I may have missed this in the beginning, but I was just wondering what is the criteria that decides whether a patient's or a donor's pancreas is going to go for research or transplantation, right? Because one of the samples you just showed was a 39-year-old male. He had no diabetes. So I'm just wondering why his islets are his islets are not used for transplantation?
Yeah, that's a great question. So to be clear, we are at the very bottom of the list in taking organs. So this organ would have been offered for clinical whole pancreas transplant or for islet transplant first, and not accepted by the clinical teams. And it's offered in Canada around the country. So it had been declined for one reason or another for clinical use. Of course, then there has to be research consent, which is separate. And once that criteria is passed, it can be offered to us. So the question then I think you have is, why would it not be accepted for transplantation? The short answer is that there are many, many reasons. One, there could be simply no suitable recipient available, with a match, like a blood type match. It could be that the time to transport the organ to an appropriate recipient was too long, right? Cold ischemic time. It could be, well, those are the main reasons. I guess there could be other reasons for clinical exclusion. But the point is that there are actually a lot of reasons why an organ may not be used for clinical transplant that have nothing to do really with the quality of the organ itself. So I hope that answers your question.
Yes, absolutely. That is what I was wondering, whether it has something to do with the quality of the pancreas or the islets.
No, actually, that's a bit of a misconception, because in most cases, there simply is not a match recipient available in the appropriate amount of time. And so that's, we can talk more about this, but, I think we're not typically receiving lower quality organs.
Yeah, absolutely.
Now, before I forget, I see that Francis Lin is here, and I gave a shout out to a couple of other people that I didn't know. I don't know if he was here the whole time, but also Francis Lin is part of this team and has contributed as well. So glad to see you here, Francis.